This is not the ideal way I'd be teaching this. Normally I do this in class. First thing you need to do before you go any further in this movie, if you haven't already gotten the worksheet, you need to download that worksheet and print it off. For those of you who don't have access to a printer, I'm sorry, nothing I can do about you having or not having access to a printer. Watch the movie if you can't print it off. Watch it several times so you get a sense of what's going on, or sketch this out for yourself and do your best. First of all, this is cell cycle part 2, and I'm just going to make my pen tip a little thinner. Cell cycle part 2. And the first thing I want to do is I want to do a quick review of why divide. You should have 3 reasons from a previous movie. The first is reproduction, because cell division like this is how single-celled organisms reproduce. In multi-celled organisms like us, there are 2 reasons. Growth, so we can get bigger, and repair. In case we have areas of the body which are damaged, or in case an individual cell had to die because it was damaged, we have to repair. So those are the reasons why cells divide. First thing I want to do is focus on this circle at the top. I'm going to just jot a line over here which should match something you have on your page. Should have a couple boxes here, CP1 and CP2. And I'm just going to note here the area of the pie graph shows the relative time a cell spends in each part of the life cycle. You can see there's one very large wedge and 2 pretty small ones, and that represents a lot of time for one and very little for the other 2. First of all, I want to identify the names of the 3 big sections. Interphase, mitosis, and cytokinesis. You might be able to fit in the whole word cytokinesis on your page. My screen shows the entire image at about 2/3 size or so, so you might be able to squeeze it in a little better than me. Those are the 3 major areas. And in the top right-hand corner, I need to write a little bit about genetic material. So first of all, the genetic material is organized in strands of DNA. Someone always asks, what's DNA stand for? Big long word, deoxyribonucleic acid. I'll say it a little slower. Deoxyribonucleic acid. Okay, be happy with DNA. And the other point, cell division requires a complete copy of DNA for both cells. So this entire process, what it has to do ultimately is make 2 new cells from 1 cell. That's what it's got to pull off. The way we're going to do this is I'm going to start over on the big circle which has interphase, and I'm going to start on the very first circle in the lower left, because interphase involves 2 big pieces. And I'm just going to turn here, so if I block the camera, I'm sorry. The first part is growth. So the idea here is this. We start with a cell with a nucleus, and the nucleus I'm going to simplify by having 2 strands of DNA. So I'm going to draw 1 short strand and 1 long strand. And I'm going to put it into a nucleus. Now I've greatly exaggerated the size of the nucleus because it's the only organelle that I'm really focused in right now. So we've got a nucleus, and the purpose of this is to have the cell get bigger. We run into this CP1, and CP1, which is right there, is CP1, checkpoint 1. So checkpoint number 1, the purpose is to check for adequate resources. So essentially the cell looks around itself and says, do I have enough stuff that I need to grow? Signals from other cells. So cells, when they're working and doing their normal thing, they're absorbing resources, they're releasing materials, and the materials they release give hints to the neighboring cells whether or not they're working properly or not. So if one cell realizes, the cells beside me are struggling to get the resources they need, they're not putting out the stuff they normally do, then that cell knows, I probably am not going to do too well at growing, so I'm not going to work too hard at it. I'm going to wait. Since I've checked and I don't seem to have enough resources, the neighbors seem to not have enough resources, let's just not do this. And they wait and they just do their normal living, do as little as they can get away with, and they wait for a better situation. Once the requirements for adequate resources are met, the growth can proceed. And so you'll notice going from the first cell to the second diagram, from this cell to that cell, the diagram has gotten bigger. The nucleus, with its 1 little strand and 1 long strand, are basically the same. The nucleus is basically the same size, but the cell around it has gotten much bigger. It's gathered a bunch of resources. That was the first thing it needed to do, was grow. The second thing it needs to do is DNA replication. DNA replication means it has to copy all of the strands of DNA. Copy all the DNA. Remember that genetic material? Cell division requires a complete copy of DNA for both cells. There's 2 strands of DNA. If we were to rip this thing apart in half, in theory, one would get the long one would get the small one. They'd have not enough instructions and the cell would die. Checkpoint 2. I'm going to do that one in orange. So checkpoint 2. The first thing it does is check to see if the DNA copied. It checks to see that the cell actually made a copy of the DNA. Kind of like a teacher checking to see if a student actually did their homework, because just because you said to do it doesn't mean it's actually been done. So the cell actually goes to the trouble of checking to make sure the copy was made. The second thing it does, no damage. or errors. Now, we want to make sure that the original didn't get damaged. We want to make sure there's no errors in the new one. The standards are pretty high. I want you to think about it for a moment. The difference in the DNA between us and a creature like a chimpanzee or a mountain gorilla are we're about 95-97% identical, is what we're told. Which means there's only a difference of about 3% between me and a mountain gorilla. So if there's 2% errors, then if the errors are right for making me into a mountain gorilla, then I'm a mountain gorilla or a chimpanzee. So 98% is nowhere near good enough of a copy. And what will happen is this. If the cell goes and checks and finds that the copying process did not go right, the cell will kill itself to prevent bad copies from going on. If it doesn't kill itself, then the bad copies of DNA get made into new cells, and depending on what those errors are, something about the new cell won't work right. So it's bad if the cell doesn't work properly, because either the cell's not doing its job, that means it's just maybe living there mooching off of the organism but not contributing anything, or worse, it's going to start doing something that harms the organism directly. If the checkpoint 2 is good, if the copies are good, if you've actually made the copy, then what we're going to see is this. We're going to see 2 short strands in kind of an X pattern and 2 longer strands in kind of an X pattern. And this time around, the nucleus is in fact bigger. The cell has stayed the same size. The nucleus has gotten bigger because the nucleus has taken in resources from the cell to make all that extra DNA. That's what's happening in interphase. Growth and copy the DNA. Those 2 things. That takes typically about 90% of a cell's life cycle. So if a fast living cell, like your stomach cell, lives for about 2 days, then out of that 2-day period of time, there will be, of the 48 hours, typically about 44 of those hours are spent just growing and duplicating the DNA. Most of that time is actually the growth gathering resources. We're now going to move on to the mitosis. Mitosis means divide the DNA. Mitosis means divide the DNA. So interphase was growth and copy the DNA. Mitosis is divide the DNA. You see there are 4 circles because there are 4 different stages. Different textbooks and resources describe them slightly differently. What I'm going to do here is match the ones in your textbook perfectly. And so the very first one, and I'm going to write their names on these, are prophase, and then metaphase, anaphase, and then telophase. A lot of people keep the order correct by using the acronym PMAT. P-M-A-T, PMAT. All right, what's going on? In prophase, there are going to be 2 things that happen. And I'm going to do the drawing first. What will happen is, gotta get the right color. So we look back at the last diagram in interphase and we see some skinny strands of DNA. And what's going to happen is the strands are going to shorten and thicken. And so DNA shortens and thickens. Nuclear membrane dissolves. So we started with a full nuclear membrane protecting the nucleus from everything else. The nuclear membrane breaks apart and the DNA gets shorter and thicker. I want you to think of those DNA strands like your shoelaces. If you take your shoelaces out of your shoes and you only have 2 of them and you just throw them on the floor, crumple them up, and then want to pull them apart, it's probably going to be okay because those shoelaces are maybe 1 meter long. DNA is an awful lot longer than 1 meter long, and they're very hard to separate if they're long and skinny. So they shorten and contract, and that way they're easier to pull apart. So that's what's going on in prophase. The DNA shortens and thickens, so they're easier to separate, and the nuclear membrane dissolves so that they can actually be separated. In metaphase, so I'm going to draw a line here separating, that was prophase. Metaphase, DNA lines up at the equator. So we're borrowing some language from geography. If I view the top of the cell as being kind of where the letter P cuts into that cell, and the bottom is out at the bottom edge, at the out end here, then what we're going to see is those strands. Remember, they've gotten thicker. So the short pair is going to look like that, and the longer pair is going to look kind of like that. And the equator line, our equatorial plane, just going to put in there's that green. So that's where the equator is. And so we've got our DNA lined up at the equator. It's more or less in the middle of the cell, so that there's about half of the cytoplasm above, half of it below. This is drawn in 2 dimensions. In a 3-dimensional cell, it's an actual layer where all the DNA line up on a flat surface. On to anaphase. In anaphase, what we see is structures called centrioles. Oh, sorry, I forgot to make it thin again. And centrioles I'm going to show as blue dots. And remember, we used that language of equator and pole. So those centrioles are going to form at the poles here. So centrioles and spindle fibers. And the spindle fibers I'm going to show in purple dashes. And go back to the green, or black, sorry, to finish writing. Form. Now I can't show the spindle fibers until I've got my DNA back in. So I'm going to try and recopy the DNA like I had in the last one. So I've tried to make them similar, short pair, long pair. And what happens is the spindle fibers start from each of the centrioles and they connect to one of those strands of DNA. DNA strands are called chromosomes. Think of those spindle fibers as ropes. Think of the centrioles as anchors, or somebody with a fishing rod. And so the whole idea is send a spindle fiber out to a strand of DNA. Now we've got one strand on each pair from opposite ends, and naturally what's going to happen is they're going to be pulled apart. So in the last diagram, which is telophase, I'm going to start with my drawing. I'm going to have my centrioles. I'm going to put those back. I'm going to draw my spindle fibers. The short one is on the left, so I'm going to put the shortest little spindle fiber I can possibly put in for each of those. I'm going to go back to my black now so I can write, so we can describe what's happening. And yeah, I'm going to put my line here separating. Okay, so what's going on? At this point, because they're separated, the strand of DNA is called a chromatid. Biologists love to keep adding names to things or changing names to things. So normally the strands of DNA are called chromosomes, but in this stage they're referred to as chromatids. So the chromatids, which are the DNA, separate. And the other thing that's going to start happening is nuclear membrane reforms. And because the nuclear membrane before was a fine line in the black, I'm going to try and do that again. So I'm going to start with the top one. And so it's going to be this loop around there, this loop around there. Now, along the way, the centrioles are going to disappear. They were only there to help separate the DNA, and once they're done their job, they're going to dissolve. Some of the materials that were making up that centriole actually go into making the nuclear membrane. So as the centriole disappears, the nuclear membrane has material to reform. And by the time telophase is all done, what we should see is, just make sure I'm on black and fine, we're moving into the cytokinesis step. Now, I'm going to write what cytokinesis is doing outside the wedge. Cell membrane. So remember, cell membrane is the outer part of the cell. Pinches off. Comma. Dividing the cytoplasm. Now, along with dividing the cytoplasm, it's also going to divide all of the organelles. So all the organelles that are floating around in the cytoplasm, they get divided pretty much equally between the two cells that are about to form. Therefore, I'm going to have a short little strand of DNA. I'm going to have a longer strand of DNA. They're a little thicker than what they'll eventually be because they were thick previously. We now have a nuclear membrane around them. We have a short and a long strand up here and a nuclear membrane around them. And what's going to happen is we see this pinching off action going in here, pinching off action going in here. And by the time we are done, what we should see is a short and long strand of DNA in there, a short and a long strand of DNA in there. And each of those two new cells are called daughter cells. And those daughter cells will now, having just finished cytokinesis, move back into interphase and want to start the process all over again of growth, DNA replication, and everything on and on and on. That's cell cycle. I've basically taken two diagrams out of your textbook and merged them together because one was going over the three phases separately and then one focused much more heavily on mitosis, so I've basically put the two of them together. I know the video has been a little long. It's been about 26, 27 minutes all total, and we would have done all of this in class. Hopefully this helps clarify what's going on during the cell cycle, and hopefully it won't have taken you more than about a half an hour to copy this, because there shouldn't have been a need for a lot of pausing and the like. Thanks for watching.